Hello, welcome to Biotanemy.
Post-transcriptional modification. The RNA is transcribed as pre-mRNA transcript from DNA. It has to be further modified into mature RNA through a process called post-transcriptional modification. There are mainly three post-transcriptional processes are involved. 1. mRNA splicing. 2. RNA capping. 3. Polyadenylation. 1. mRNA splicing. The pre-mRNA transcript contains two regions called introns and exons. Intron or introgenic region is any nucleotide sequence within the gene that will not express in the final RNA product. And exon or expressed region is the part of the gene that will be seen in the final mature RNA. The introns are removed from the final mRNA transcript through a process called splicing. For example, consider the pre-mRNA transcript with two exons and one intron. The intron contains important regions like 5-GU sequence or 5-splice site, a branch site, that is located near a pyrimidine-rich region, and 3-AG sequence or 3-splice sites, that are required for splicing. The splicing process is controlled by a large protein complex, which is made up of RNA and protein, called spliceosome. These particles are called small nuclear ribonucleoproteins or SNRPs. The SNRPs will identify the above-mentioned conserved regions seen in the introns and bind to them to initiate splicing. The spliceosome will form in the pre-mRNA transcript in the following sequence. The SNRP U1 and U2 will bind to the 5-splice site and the branch site respectively, followed by the binding of SNRP U4, U6, and U5, respectively, to form a mature spliceosome complex. This causes the intron to form a loop-like structure to bring the 5-splice site and 3-splice site together, to begin the splicing. The 5-end of the intron will be cleaved first. The free 5-GU end of the intron will be connected to the branch site A, to form a lariat structure. During this stage, the SNRP U1 and U4 will be released from the spliceosome complex, and 3-splice sites will be cleaved. The lariat form of intron along with SNRP U2, U5, and U6 will be released while the exons are attached. The spliced introns will be degraded and spliceosomes will be recycled. 2. RNA capping. In order to prevent degradation of mRNA, the 5-end of mRNA is modified by adding 7-methylguanosine residue in a 5-to 5-triphosphate linkage. The mRNA cap will recruit ribosomes to mRNA to promote translation. The mRNA cap may be added during transcription, including the cap analogs, or after transcription with the use of capping enzymes, GTP and S-adenosylmethionine or SAM. 3. Polyadenylation. The process of continuous addition of adenine to the 3-end of mRNA is known as polyadenylation. The polyadenylation begins by cleavage of free 3-hydroxyl from the 3-end of the mRNA transcript. Then the enzyme poly-A polymerase will begin to add adenine nucleotides to the RNA. The poly-A tail addition will continue up to 100 to 250 residues long. The poly-A tail will prevent the RNA molecule from degradation and make it more stable. It will also help to export the mature mRNA molecule from the nucleus to the cytoplasm.
Thank you. Please like, subscribe and share. See you again in a new topic.